The question is, are there human cells which have properties similar to the human embryonic stem cells, but are not derived from the embryo? Well, fortunately, the answer is yes. In 2006, a Japanese scientist named Shinya Yamanaka has discovered these cells, and he named them iPS cells. So I will give you an example using iPS cells, treating a mouse model with sickle cell anemia. So again, just remind you that traditionally, pluripotent stem cells are derived from the early embryo. And what Yamanaka and colleagues did differently is that they took skin cells from adult human and then introduced 4 genes into these skin cells. They carefully picked these 4 genes so that they're capable to revert the development of these skin cells into their early development stage. And this process is known as reprogramming. And the cells derived in this way are known as induced pluripotent stem cells, or iPS cells. Yamanaka and others have shown these iPS cells are similar to embryonic stem cells. They're able to give rise to all possible cell types in adult animals. So this discovery has revolutionized the way we think about treating diseases in an individualized way, because this approach provides a reasonably easy way to make pluripotent stem cells for each adult patient using their own cells that are very easy to get, such as skin cells or peripheral blood cells. So 1 year after this discovery, a group of scientists at MIT have used these iPS cells to treat a mouse model with sickle cell anemia. So they took skin cells from mouse carrying sickle cell anemia and reprogrammed them into iPS cells. And because we know the mutation that caused sickle cell anemia and its exact location in the genome, they were then able to correct this mutation by gene targeting to make genetically corrected iPS cells and differentiate them into blood stem cells and transplant into the sickle cell mouse. A few months after the treatment, you see mouse recovered from the illness. So these similar approaches have also been used on animal models with other human diseases, such as Parkinson's disease. And these animal studies are very promising. And this is still a relatively new field. So far, no one has tested this in the clinic on human patients yet. But many people believe these iPS cells are the ultimate future of personalized medicine. Imagine the future a patient comes to the clinic, and his doctor will be able to take a skin biopsy from a patient to make pluripotent stem cells for this individual patient. And if the disease is genetic, and if we know the mutations that cause the disease, the doctors might be able to genetically repair this patient's specific stem cells and then differentiate them into liver cells if the patient has liver disease, to replace the bad cells and transplant these iPS cells, these liver cells, back to the patient to replace the bad ones, thus cure the disease. On the other hand, if the disease is not very well studied and we don't really understand the cause of disease, these patient-specific stem cells can be used to study the disease in the laboratory. In addition, the doctors will be able to test all the available drugs using these patient-specific cells in order to find the right drug for the right patient and prescribe him with the right dosage. So there are still a lot more work that needs to be done before iPS cells can be used in a clinic. So the current major limitation is that these cells are very difficult to obtain. So the reprogramming efficiency is very low. On average, people have to look into the microscope to screen for about 100,000 colonies in order to find out one colony which have been successfully reprogrammed. And the most efficient way to make these iPS cells is to use a special vector which stably integrate those 4 genes into the genome. As you can imagine, this might cause some unwanted mutation, having some deleterious health problems. So scientists have to develop a more efficient and non-invasive way to make these iPS cells in the near future. Furthermore, currently there's this ethical debate. Currently there's this big debate about whether iPS cells are as pluripotent as embryonic stem cells. So in many studies using iPS cells, human embryonic stem cells have often been used on the side as a comparison. So unfortunately, at this point, we're still unable to totally abandon research using human embryonic stem cells. Moreover, there is always a concern about safety. The cells that are going to be transplanted into the patient have to be highly purified. Any contamination with undifferentiated stem cells will have a potential to cause tumor. Finally, despite all these years of research on stem cells, we're still unable to obtain certain cells using these embryonic using stem cells. And this is very important because one can use stem cells to treat diseases such as type 1 diabetes because we know how to differentiate stem cells into insulin-producing cells. So similar procedures needs to be developed for other cell types in other diseases. So in summary, stem cells play a very important role in the future of personalized medicine. Adult stem cells have no problem with, doesn't cause problem of immune rejection, but these cells have limited potency and they are present at very small quantity, very difficult to get to large enough quantities for therapeutic purposes. In contrast, human embryonic stem cells, in theory, can provide unlimited quantities of all cell types. But because they do not exist in adult patients, one has to use cells from a foreign source, and this causes problem of immune rejection. And lastly, these patient-specific iPS cells not only can provide unlimited quantities of all cell types, but they have no issue of compatibility because these cells can be derived from patients themselves. And the current limitation in regarding to these iPS cells are that they're relatively inefficient to produce and have very high maintenance. So tonight, we've told you about several recent important discoveries in the field of biomedical sciences and how these discoveries changes our view on treating diseases and led us into an era of personalized medicine. Monica has told us about the human genome and how we could use the information encoded in our genome to predict the risks of getting certain diseases, thus prevent the onset of certain diseases by changing our habit, for example. Yixiao has told us about how the genetic information encoded in our DNA can be used to diagnose illnesses, and how pharmaceutical companies can use that same information to develop better treatment for diseases such as breast cancer. And we've also highlighted some of the important discoveries in the field of stem cell biology, and how stem cells can be used in the future of personalized medicine, and where all these discoveries lead us to in the future, and when and how they will be incorporated into the healthcare system are unknown. But one thing I do want to point out is that the future is not only in the hands of scientists, but also determined by you guys, everyone sitting in the audience today. So with that, I would like to thank you all for coming tonight, and we'll be happy to take any questions.